So good afternoon, everyone. Welcome to this edition of the NITheCS Colloquium. My name is Francesco Petruccione, and today I have the great pleasure of introducing Professor Hugh Patterton to you. Hugh is currently the director of the Center for Bioinformatics and Computational Biology, so he fits perfectly the profile of a NITheCS associate. So welcome to our NITheCS club, Hugh. Hugh studied biochemistry at UCT and received his PhD there in 1991. Then he moved for a short while, almost 10 years, to the US, on various stations there that I won't read or printed on your announcement. But he returned to South Africa in 1998. With various stations at University of Free State, at UCT, back to University of Free State, and eventually to Stellenbosch. Maybe I got it wrong now, but he was fundamental in setting up the national bioinformatic networks to establish bioinformatics in South Africa. And today he will give us a personal account of his bioinformatics journey. So Hugh, we are really very happy that you are with us today, and we are looking forward to your presentation. And while you share your screen, I quickly remind the participants to use the Q&A to ask questions, and maybe after the talk we can allow participants to raise their hand and ask questions in person if they so wish. So Hugh, please, over to you.
Good. Thank you very much, Francesca, for your very kind introduction and invitation to speak to you today. I'll take it that my screen is visible to everyone.
Yes, it's clearly visible. Very much.
So when Francesca asked me for a title, I thought, well, I'd like to perhaps chat a little bit about bioinformatics, but sort of center the bioinformatics a little bit more on some of the biological questions that I've been asking as a scientist. And hence the qualification of personal journey. I'm not going to have so much as a raconteur's exposition of my progress through a specialized field, but rather sort of just cherry pick some of the topics in bioinformatics and show how I applied it to my own research. Okay, so what I'd like to kick off with is if this can next, there we go. Okay, I'd like to kick off with is just show a tree of life based that's a little bit more modern than some of the ones that one would see, based on genome sequences. And there are a couple of things that I'd just like to lift out here. First of all, that there are three large kingdoms: the bacteria, the archaea. Let me just get my laser pointer going here. The archaea, and then the eukaryotes. In terms of the eukaryotes, there are two important points that I think that one should be aware of, and that is this is the last eukaryotic common ancestor, LICA. Before the last common eukaryotic ancestor, the basically evolutionary line took up an alpha proteobacteria, which evolved or developed into what we know today as the mitochondrion in a eukaryotic cell. And some time later, evolutionary-wise speaking, it also took up basically a cyanobacteria, and that was utilized and is being utilized in plants to photosynthesize and actually allow us to use solar energy to reduce sugars or to synthesize complex organic molecules in which we can store energy. We can use for both building blocks or use for eventual re-breakdown to release the energy. So this is just a simplistic representation of that. And if we actually look at some important events in the history of life on this planet, then you'll see that the first eukaryotes probably appeared about 3 billion years ago. 2.5 billion years ago was the great oxygenation event, during which there was a significant increase in oxygen in the environment of the planet. Most of that was not available. It was basically reacted chemically and formed various types of geological deposits that one can still see today. And it was only about 1 billion years later that we actually had free oxygen starting to appear in the Earth's atmosphere. And this was followed by the acquisition of a organelle that's known as a mitochondrion. And whether this was prerequisite or simply happened before the appearance of the first multicellular organisms is not quite clear. But we know that we have the first metazoans only appearing some time after we've developed the ability to use oxygen as a terminal electron acceptor in the propagation of electrons and protons across the inner mitochondrial membrane to generate high energy molecule, in this case ATP. It was sometime later that the first land plants and animals appeared. And something that was quite interesting is at the Precambrian or very early Cambrian, there was an enormous explosion of the diversity of organisms. And this is known as a Precambrian explosion. And actually, if you look at the fossil record of the Precambrian explosion and look at the distribution of different organisms from that period, then you will see a representation of virtually each and every phylum that we can analyze, that we can identify today. So it's actually quite extraordinary that this reasonably short period of time, we had the development of virtually all the different groupings that we can recognize today. The reason for that may be manifold. It may have to do with the development of the way in which organismic development took place in terms of specific genes such as the notch genes, etc., involved in laying down proton, basically concentration gradients and formation of and the distribution of cell types in the formation of organisms. And that this made it much easier to actually experiment and with a large variety of different forms, which led to the early Cambrian explosion. That is but one hypothesis. There are many other hypotheses as well, but certainly we do see that tremendous increase in the number of identifiable phyla during that evolutionary period. Over here is just another tree that I'm showing you is from Wusa. Those of you like myself who do suffer from a little bit of astigmatism will probably be slightly upset by the appearance of the center of the tree, because it looks a little bit like the typical test for astigmatism. But I can assure you that the visual lines are actually perfectly focused. And the purpose of this tree, what Wusa tried to show here, was the proposal that eukaryotes can possibly be thought of as a basically mixing of the genetic background of bacteria and archaea, and lateral gene transfer between some of these individual lines, and that this is a reasonable way to actually think of a modern eukaryote. Okay, so having looked a little bit or spoken a little bit about the development of the different groups in the tree of life, the next thing that I want to basically mention is the fact that if you look at the chemistry of life and you look at the chemistry in a bacterial cell or in a eukaryotic cell, there are more things that are the same than things that are different. There's specific groups of chemical reactions. So the one would, for instance, be the breakdown of a sugar molecule, such as glucose, and the feeding of bits of that sugar molecule into a cycle that's known as the citric acid cycle or the Krebs cycle. And this spins off basically carbon dioxide and generates reducing power in the form of NADH, which allows the molecule to basically deliver electrons and protons to the inner mitochondrial membrane and generate energy molecules such as ATP. But it's not only an ATP generating precursor cycle, it is also a cycle involved with the manufacturing of complex organic molecules that is used in the construction of the cell. So, for instance, there are links through two chemical reactions involved in the biosynthesis of nucleotides or amino acids. It's not necessary for the talk here to go into the detail of that, but simply be aware of that. That this grouping of chemical reactions also share common reactions that are involved in fatty acid biosynthesis or the breakdown of fatty acids for energy. So what I'm trying to communicate with this image is simply that the cell is a chemical system. There are many, many chemical reactions taking place. Some of those chemical reactions have to do with the generation and capturing of energy, and some of them have to do with the manufacturing of complex molecules that can be used as building blocks in the construction of a more complex physical entity. Okay, so I think that's something that hopefully would have immediately became obvious in the previous slide, is that if you think of a cell in terms of a chemical system, then invariably you start thinking of a von Neumann automaton or automata. That the chemistry of the cell is basically equivalent to the manufacturing part of the von Neumann automata. And something that I haven't mentioned up to now is that the cell will also contain information, and we'll be looking at that in a second, that is represented by the information ribbon here in a von Neumann automata. And the point here is that this contains all the information to allow the construction of the automata. The automata itself can basically recover resources to construct a copy of itself. And there's also a replicator that allows the replication of the information ribbon to basically duplicate the entire von Neumann automata into a total new copy of itself that in this case would be identical to the first copy, and this copy can now continue and duplicate again. And hopefully the similarity between such a type of system and a living cell is quite obvious. And if you overlay that now at a simplest level, just by using a genetic algorithm. So over here, obviously this is now an in silico approach. It's not a chemical approach, but I think the concept here is the same. That essentially what you would do is you mutate the information in the living cell, and then you'll go through a process to evaluate the fitness. So this is just pseudocode for a genetic algorithm. You evaluate the fitness of the mutated cell machine or cellular automaton, and one can actually apply this type of genetic algorithm, as I'm sure many of you have done, on something that's quite simple. For instance, you can use it just to mutate and test and select the best functioning versions of a neural net that controls something like a simple game. In this case, it's just the game of Pong. And So this game, as I'm sure all of you would know, is simply a tennis ball that basically bounces against the back and side walls of this hole, and then basically progresses. And the idea is that it should hit the paddle to keep it from flying out of the court of play. And the point of basically using a genetic algorithm is just to select the versions of the neural net containing parameters that performs best. And you basically mutate that, and you keep on selecting the ones that perform the best. And typically after a couple of hundred or after 1,000 generations, you come up with something that plays the game almost perfectly, that it will never lose the ball. And it's always been a point of puzzlement for myself that, for instance, in high school biology, the whole concept of genetic algorithms used at selecting specific sets of parameters and then testing the parameters for specific advantageous characteristics is not used much more often in teaching biology and evolutionary concepts in biology. But I think the point here is made that giving a cellular system, it is possible to basically tweak or change or mutate some aspects of the information that will have a knock-on effect on the chemical system itself, and that one can select the chemical system in terms of its performance of a specific series of activities in the environment which is deemed to be advantageous for that particular system. Okay, so if I take just a brief side step here and just point out that if you look at these chemical systems, then sometimes you hit a little bit of a problem, especially for the advanced chemical systems such as the ones that we can see in existence today. And that is, yes, you do have the chemistry. So over here I have 3 metabolites that are connected by arrows. However, if left to their own devices, it's likely that these chemical reactions will proceed extremely slowly. And it may well be that there are side reactions that run more quickly than the reactions that we've indicated here with the blue arrows. And the side reactions would be reactions that we do not specifically want. So typically, to speed these reactions up, we need to catalyze them. We need to basically lower the activation energy of the chemical interconversions. And to do that, we typically make use of biological catalysts, otherwise known as enzymes. So the enzymes are these guys over here. And the intellectual problem comes in that the enzymes are originally coded for by the DNA. This is the information molecule, right? So the DNA has to be transcribed into mRNA, and the mRNA then has to be translated into a sequence of amino acids, and this occurs via complementary binding of tRNAs that contains an amino acyl group, which is the part of the amino acid, and that the specific order of trinucleotides in the mRNA dictates the orders of the order of the amino acids in this growing peptide, and the order of the amino acids in the peptide will basically govern the folding of this protein into a specific catalyst that can catalyze a specific reaction. So the problem is that it's extremely unlikely that this type of system will evolve in the absence of this system. And typically, and also a chemical system such as this, giving rise to the evolution of a system that catalyzes specifically the described system. So this is basically chemistry first versus information first. And at this point, it's not entirely clear how to arrive at a solution to this. There were some attempts, for instance, the molecules known as self-cleaving ribozymes. So in this case, you have a ribozyme that will basically self-cleave its phosphodiester bond to form a cyclic phosphodiester, and a second part, so basically the molecule itself is hydrolyzed at the phosphodiester bond. And this is a self catalysis. So obviously you can have a molecule that in principle can act as an information molecule, but at the same time is also a catalyst. The only problem is, how do you actually get the catalyst and information synchronized so that you can select for something that contains the information that you actually want to select for the propagation of a specific chemical reaction within your chemical system. So I think this is not yet entirely clear, and this is an area that is actually quite actively being inquired into, and which I think is actually a fascinating area. Okay, but I think that that having said all of that, we will probably agree that one can view a living cell as a self-replicating machine. It's a type of a Neumann automata. And I think it's particularly helpful to think of living systems as von Neumann automata in trying to dissect the various functionalities of that system. So over here, I've just listed 4 possible sources of energy. So not all life on Earth now requires sunlight. Initially, obviously, since we acquired cyanobacteria some time after the origin of life, there must have been a period when the chemical systems actually acquired energy from sources other than the splitting of water with energy from sunlight. And here are some examples of some of these energy sources, some of which are still in existence today and utilized by specific archaea and bacterial species. Okay. So the point up to here that I wanted to make, that is that I want you to carry forward, is that a cell should be seen as a self-replicating machine, as a type of a Neumann chemical automata. Okay, so if we now take a step back and think in terms of the information molecule in the cell, so that is the genome. The genome is basically the entire complement of DNA, or RNA for that matter, in case of an RNA encoding system. The total encoding essentially of all the information in the cell. So over here is a plot that that shows the 3 kingdoms of life, and the blue bars, which looks a little bit like a planet with some high rises appearing on the surface, and the gray sectors basically represents the bacteria, the green sector the archaea, and the pink sector the eukaryotes. So the height of the blue bar indicates the size of the genome of the organism. So I think something that's immediately striking is that it seems that bacteria seem to have some type of either maximum size, or that in terms of the complexity of the system, they don't need a genome larger than the genome that is present in most bacterial cells. And if you look at the archaea, you also see that there certainly seems to be some type of limit, either functional or selected by the ability of the cell to maintain that system, that is comparable in size to that of bacteria. If you look at the eukaryotes, on the other hand, you see that suddenly the genome sizes absolutely exploded. So there's something different between bacteria and archaea on the one side and eukaryotes on the other side, that allow eukaryotes to basically maintain these genomes of these sizes, or require genomes of these sizes. And there may be a little bit of a truth in both of those constraints. So it's probably a good idea just to stop for a second and look at the amount of information that is in DNA. So I've been quite conservative and basically stated that every base pair can be encoded by 4 bits. So all of you will know that there are 4 different nucleotides, J, T, and C. So you only need 2 bits, or 00, 10, 01, or 11. So 2 bits to encode the identity of one of the 4 nucleotides. But apart from the chemical signature of the nucleotide GOT or C, there can also be modifications. Some nucleotides may be methylated, some may be glycosylated, etc. So to allow for that little smattering of additional modification states of the nucleotides, I've specified that that each base pair can be represented by 4 bits of information. So the human genome is about 3 billion base pairs, which means that it can be encoded by 4 times 3 times 10 to the 9 bits of information. And that's about 1.4 gigabytes of information. So the astonishing thing here is that the entire human genome, the entire information molecule that encodes the mechanism required to generate the chemistry in cells that make up humans, can be stored in only 1.4 gigabytes of information. The other astonishing thing with this is that this amount of information is contained in a cell with a cell nucleus. So in eukaryotes, the eukaryote has a nucleus. Archaea and bacteria do not have a nucleus. In eukaryotes, the cell nucleus only has a diameter of approximately 10 micrometers. So if you actually take all of the chromosomes in a human cell and you unravel it and lay the DNA or lie the DNA end to end and end it up, you would find that in a human, you have approximately 2 meters of DNA in every somatic cell. So since each human is composed of about 1 trillion cells, that means that each human contains about 2 times 10 to the 12 meters, or 2 billion kilometers of DNA. The distance from the sun to the planet Uranus is about 2.8 billion kilometers. That means that a single human being contains enough DNA to stretch from the sun almost to the planet Uranus. So that is actually quite astonishing. And the one thing that I think that particular fact should really underscore is that there's something that's happening in a human cell that allows it, or even in a eukaryotic cell, that allows it to fit something like 2 meters of DNA into a cell nucleus of only 10 micrometers in diameter. And that thing that happens, in fact, is chromatin, and that's the packaging of the DNA onto a protein complex. So over here is what is known as a nucleosome. So it's composed of histones. These are the colored little spirals in the center, and the gray area on the outside here is the DNA duplex that is spooled in about 2 negative superhelical turns onto the surface of the histone octamer. So the histone octamer itself is composed of, as I said, histones, which is composed of a typical histone fold motif, which is a short helix with a turn, a long helix, and a turn, and another short helix. And you'll see that each of the different color histones actually have the same type of arrangement, known as the histone fold. And they basically associate in these dimers. And the blue histones here are known as histone H3, and they associate with a 4 helix bundle in the central part of this nucleosome. And then there's H2A, H2B dimer over here, which is the yellow and orange part. And this associates with also a 4 helix bundle via the H2B domain with the H4, the green molecules histone H4. So you basically have a 4 helix bundle here, and you have another 4 helix bundle here, where the dimers basically associate to generate a surface onto which the DNA can be spilled as a negative supercoil. In the case of Archaea, you have something that's quite similar, although the eukaryotes have 4 different histones. In fact, we have more than 4, because we have various histone isotypes as well that are expressed in specific tissues or at specific stages during the cell cycle or during development. But I won't be going there for today's talk. In the case of the Archaea, the histone equivalents are much simpler. Although unmistakable, you can see the type of histone folds structure appearing in the archaeal histones. Okay. So the other important thing here is that if you look at the histones over here, the purple disk here, if you look closely, is in fact composed by two purple disks. And you have a negative superhelix on the one purple disk, and then it goes over to this side and continues as a negative superhelix on that purple disk, and then it comes over to the orange in the center, etc. So you basically have the arrangement of the DNA onto these disks, and they run between the stacks of disks. And each stack basically forms a negative helix as well. So if you look at the pseudo-atomic model of this, you'll see that the DNA is in fact arranged as two helical ramps, two start helix, with the DNA running between the stacks, nucleosomal stacks within either of these helices. So this arrangement allows significant compaction of the DNA to actually fit into the cell nucleus of only about 10 micrometer diameter. The difference between the eukaryotic chromatin and that of archaea is whereas over here we have these two start helix of interwinding helices. In the case of archaea, the DNA is basically wound onto a central rod that's formed by the archaeal histones. So you can see immediately the degree of compaction that's feasible or possible in archaea is much less compared to that of eukaryotes. So the point is, in a eukaryote, do we have larger genomes because we can accommodate that, or do we need to accommodate the larger genomes because we have to encode more information to form a eukaryote? So the cause and effect there is not entirely clear, but this is certainly an important part for understanding why there is such a difference in the size of the archaeal and eukaryotic genomes. Okay, so if we just forget the histones for the time being and just quickly look at the DNA and the functionality of the DNA. So this is just a very simplistic representation of a gene, a eukaryotic Pol2 transcribed gene. So you'll have a title box and you'll have the whole pre-initiation complex that will be assembling on this part of the proximal promoter area. There can be some additional transcriptional activators that are present on the proximal promoter and the distal promoter that stabilizes the formation of the pre-initiation complex here. And then this entire region is basically transcribed by RNA polymerase II, and then the RNA is processed and the introns are spliced out to actually form a mature transcript composed of the exons that is used for the translation into or encoding the sequence that is used of codons used for translation into a series of amino acids to form a protein. Okay, so this is the type of information that one would often want to extract from DNA to actually gain some insight into the functionality. And to get that type of information, you have to be able to get at the DNA sequence, right? Although nowadays there's tremendous amount of genomes available on internet and various databases, we had to generate the sequences by some means, and we're continuing to generate sequences of unknown genomes still. So if I can just take a minute quickly to explain how we're actually generating those sequences. The first part would be essentially to generate a library of fragments, and then to the ends of these fragments, you basically attach a type of adapter. I won't go into the detail of this, but the adapter itself is designed so that once it's attached to the fragment, that you have different sequences on the strand. So you can see here on the right-hand side, you have a blue line, and over here on the left-hand side, you have a green line. So that is very, very important because that allows you to bind either the one side of that strand or the other side of that strand. How we actually sequence the insert now is that we anneal the fragment that contains the single-stranded adapter at the end to a surface, and after having aligned it to a complementary oligo on the surface, we extend the oligo, we synthesize the complementary strand, and then we can basically wash off this original template strand. And the important thing to amplify now is that we do it by bridging amplification. So we allow this newly synthesized strand to reach over and essentially anneal, in this case, the blue adapter to another blue sequence, and we extend the blue sequence using this bridging molecule now as a template. And by repeatedly doing this bridge amplification, we're actually amplifying up a little forest of DNA strands, and they all have the same sequence, okay? But they either attached to the green adapter or to the blue adapter. The initial density that we use to actually start the bridging amplification should be such that these forests are sort of isolated little clumps on the surface area. So we don't want the forest to sort of flow into one another. We want them quite distinct, physically separate on the surface of the slide. Once we have these forests, one can actually then use a primer, and you can synthesize the template strand again. And every time, so remember all these molecules, all these fragments within a single forest on the slide are identical, okay? And you first remove all the ones that are attached with the green adapter, so you only end up with the ones that are attached by the blue adapter, and you sequence them. And since they are all identical, and when you basically incorporate a specific nucleotide, it contains a fluorescent marker. So this slide over here represents all the individual little forests, and you can see that each forest will fluoresce with a specific color depending on which nucleotide was incorporated into the growing strand being synthesized, matched on the template strand. So each individual forest are sequenced simultaneously, but it's independent, obviously, because it's a physically independent structure on the slide. But this parallelizes the whole process. So you get all these sort of colored dots appearing, and as you continue to incorporate the nucleotides, you will build up the sequence of each forest by the color of that fluorescence that you observe. So this is the basis of cyclic reversible termination sequencing, which is something that is used on the Illumina platform. This is just once again showing the individual spots and the synthesis of the growing strand. And here's the fluorophore, and there are various chemicals that are used as fluorophores here that fluoresce at different wavelengths that one can observe. Okay, so the whole process here is parallelized, so we can actually sequence hundreds of thousands or even millions of molecules simultaneously on such a sequencing platform. The file that is generated by the sequencer is something that is known as a FASTQ file. So it's basically composed of blocks of information. First line here would simply be the information of that particular forest, the sequence itself. Then there's space here for some additional information. And then the fourth line gives you the quality of the assignment of the nucleotide at each position. Okay? And then you can see that this occurs in blocks of 4. And over here is the next block, so the next sequence, next block, next sequence, next block, next sequence, etc. And this is just a teeny weeny bit of a FASTQ file. So these files will typically be gigabytes in size that one will recover from a DNA sequencer, and that one would have to process to actually gain any insight. Okay, so one of the first things that one would do if you actually had an unknown, so you had a bacterial cell you isolated from some type of ice valley in Antarctica, and you're interested in assembling a genome, you essentially sequence the fragments from the genome of the organism. And the point is, how do you actually put this together now to actually recover a genome sequence that you can actually work with and say, well, this is the genome encoding all of the information for this particular bacterial cell. What you essentially do is you set up a hash table where the individual reads are hashed, and then you basically find reads that overlap. So for instance, over here, so I'm using very, very short sequences here just to demonstrate the facts. We use much larger sequences to actually do the proper genome assembly. So over here, terminating in the 4 nucleotides ATT. So what you'll do in the hash table is find something that starts with ATT. And then if you're lucky, you'll only have one molecule that starts with ATT over here as a 4th nucleotide A. So the next one, you have to find something that overlaps with TTA, next one with TAC, etc. If these sequences are too short, then they're going to not be particularly unique, which means that basically at every position, you're going to have probably 4 different nucleotides that fit. So basically it fits the overlap, but then there can be 4 different nucleotides overhanging, and you don't know which one is necessarily the correct one. Also, if you have duplications, that would mean that there is some type of duplication within the sequence itself. So after assembling this, you essentially generate, or what a program will do is generate a brain graph. And in this case, the Eulerian walk through the brain graph is basically the sequence. That's basically but typically it doesn't work out quite as tidily and easily as that. There are rounds of additional tweaking that you have to do to actually get to the final sequence. But this demonstrates the basic principle. And over here, for instance, if you have a CTAC, TAC over here, basically duplicating this particular sequence, so that means that you basically have an area that folds back on itself. And this is quite simple. So you know that, well, it has to go around this way once and then follow this route the second time. So that makes it easy. But sometimes you actually have quite complex junctions, and it's not quite obvious how the path should proceed through all of this. But anyway, this is the basis for actually using these short sequences to basically overlap them and then put them together and try and identify the Eulerian path through the brain graph, which then represents the sequence itself. So here's an example that I always give the third-year students, just to show to them that, well, this works. And this is just taking 500 nucleotides and just generating or randomly selecting 50 nucleotide sequences from this after 50-fold coverage. So if you stack it, you have basically about 50 copies of sequences at any position, and then taking it through a program that basically assembles it by this partial overlap. And eventually, this is the brain graph, which is basically just a perfect Eulerian walk, or simple Eulerian walk through the assembly. So this is the simplest representation, but just to show that the process in fact works. And this is a major way in which genomes have been assembled. We're actually seeing additional techniques coming in now because we have new types of sequencing technologies that have come to the fore, and including the packed biotechnologies and the nanopore technologies that allows us to sequence very, very large pieces of DNA running into even tens of kilobase pairs. which changes the story somewhat. Okay, I'm going to jump over that and just quickly, because I see I've used up 45 minutes, and basically just mention that quite often when you try and find sequences, so if you go to a word processor, something like Knuth-Morris-Pratt is perfect, where you will basically have one pattern and you'll basically construct your prefix, suffix, prefix connection and the file table, and use that to basically search the sequence. The problem is that if you have a read, such as that read in the FASTQ table that I showed you, and you're trying to find its location in a genome, yes, you can use the Knuth-Morris-Pratt algorithm, but not if you have many reads, because then for each and every read you have to make a new skip table, and that makes it really, really inefficient. So an algorithm that we've been using that works really well is the Burrows-Wheeler transform, or the Burrows-Wheeler algorithm, that is based initially on something that was designed in compaction. And how this algorithm works, I'll just take a minute to very quickly run through it, is it just produces a rotating permutation of the sequence. So in the real world, this top line will be the entire genome sequence, okay? And basically generating a rotatory permutation of that sequence, and then lexicographically sorting it from the first column. And then the last column in this matrix is the Burrows-Wheeler transform. And the Burrows-Wheeler transform has the interesting property that the rank order of a symbol in the first column, so over here the symbol A, first one ranks 0, second one is rank 1, and third one's rank 2. The order here, the rank order is the same as those symbols in the last column. And the reason for that is because they are lexicographically sorted on this symbol, and then this symbol, and then this symbol, and that's the same as on this symbol, and this symbol, and this symbol. So the symbols on which they are sorted basically are the same. That's why the rank order in the first unlined columns are the same. And how this can be used to basically find a sequence, for instance, if you're trying to find the pattern TTAT, you only have to generate the, actually just the Burrows-Wheeler transform. You don't need the matrix. I won't go into how that works in this lecture, but essentially taking the transform and you're looking for the sequence TATT. So what you will find is, or what you will follow is a sequence T that terminates in A. So over here you have an A, and this was preceded by, I beg your pardon, this is a T and was preceded by A, and this guy was also preceded by an A. So the rank order of the A here, so there's an A up here. So this is 1 and 2. So you can see over here, this A was preceded by T, but this guy was preceded by G. So you can ignore this, and you can follow this guy. This is the first order T, and you can see that this T was preceded by another T, and there you found the entire sequence. So basically by jumping between the last and first, or by using two tables known as a seed table and a occurrence table that only the Burrows-Wheeler transform, one can actually find, very rapidly find short read sequences in the original text. And you only need to generate the transform once, and then you can just sort of step your way through all these patterns and find them. So it's been a very, very efficient way of actually looking for matching short reads, say, to genome sequences. And I think that that we're on 50 minutes. So I think what I'm going to do is actually at this point stop, and I think I've said most of what I wanted to say, and invite any questions from the floor, or Francesco, if you have any specific queries that you have.
Thank you very much, Hugh, for the excellent introduction to bioinformatics. Really very nice.